Today's speaker is Dr. Tom Polasek. Tom is a clinical pharmacologist at Certara. His research interests include all aspects of clinical pharmacology with a focus on the clinical application of modeling and simulation approaches. He is the author or co-author of more than 80 peer-reviewed articles and conference presentations. Tom earned his Bachelor's of Pharmacy degree from the University of South Australia and his Bachelor's, Medical degree, and PhD degrees from Flinders University. Tom, welcome to today's event. I'll now turn it over to him to begin the presentation.
Well, thanks, Suzanne, for that really kind introduction, and thanks everyone for joining the webinar, either watching live now or those who are going to be watching and looking at the slides at a later date. Okay, well, today I want to talk about this incredibly exciting world of model-informed precision dosing, which I abbreviate as MIPD, and the focus of the talk is virtual twin, so how we generate a virtual twin of an individual patient and how they can be used to predict drug exposure. So the talk today comes from a very clinical perspective. So what I want you to do is imagine yourselves as one of the healthcare team that's actually looking after a patient. So the potential end user of any MIPD technology that's developed. So for those of you who don't look after patients regularly or who have never looked after patients, I want you to get out of your comfort zone a little bit and think about what it would be actually like to use a technology in a healthcare setting. So what I've got is I've got 3 clinical cases during the talk where I'm going to ask, if you were responsible for looking after this patient, do you think model-informed precision dosing would have a role in improving the quality of their care? So here are my disclosures. I work half-time for Certara and half-time for SA Health here in South Australia. So for those of you who like to monitor progress as we go through the talk, there are 5 main sections, 70-odd slides. So in the background section today, the first case is an elderly man who's got complex medical problems and is on polypharmacy. And I want you to ask yourself whether model-informed precision dosing could be used in that individual's care. Then I'll talk about the different approaches to MIPD. There'll then be a little overview of virtual twin concept and how the models that have been developed so successfully in drug development could then be expanded into healthcare. The second part of the talk is about psychiatry, and the second case is a new psychotic illness that's treated with olanzapine. And as part of that section, I want to talk through the proof of concept paper for virtual twin that we've recently published in the BJCP. Then we move on to the oncology section where we meet a man in case 3 who has something called gastrointestinal stromal tumor, and he has very bad imatinib toxicity despite having really good disease control. And what I want to show you during that section is how the team at Flinders University, led by Andrew Rowland, are using Simcyp to discover covariants that could be really important in predicting exposure to kinase inhibitors. Then I'm going to highlight some advantages of a virtual twin, including potential large cost savings in particular cases, and then the conclusions, which is then going to be followed by the question and answer session. Okay, so before we get into all that, this is my definition of MIPD. Just reading off the slide, it's by simulation in healthcare to predict the drug dose in a patient that is most likely to improve efficacy and or lower the risk of toxicity. So some key points here, it must be in a healthcare setting. We're currently focused on one drug as part of someone's care and its dose. We're focusing on one patient, a given individual patient that you're looking after. And you can see here it covers all types of responses, efficacy right through to all the toxicities as well. So I haven't defined the end user, but it's most likely going to be the prescribing doctor or a clinical pharmacist who takes leadership in this area, but not necessarily so. That isn't defined at the moment. Okay, this is our first case. So let's talk about this gentleman, an 82-year-old man who comes from home. Unfortunately, his wife died last year. He's largely independent, though. He's been referred in by his general practitioner or family physician. He's had repeat hospital admissions in the last 6 months with several falls at home, and it's just called acopia on the referral. Here's his medical history. There's quite a lot going on there, and here's his long list of drugs. So you don't have to go through and count all of them. I can tell you that there's 16 medications on here, and surprisingly, this is actually not rare in clinical practice. So as the doctor looking after this gentleman here, you've got 1 hour to read the referral note, look at the background information, look at discharge summaries, some imaging, pathology reports, these type of things. You've got to take further history from this gentleman. We have to examine the gentleman and document everything really well, make a problem list with some priorities, got to formulate some plans, and then start the necessary follow-up. So with all that to do, here are the problems that we come up with in terms of a ranking. We were actually really worried in this gentleman that the cause of his falls is some critical lower limb ischemia, which is a medical emergency. And you can see he's got some other very serious things going on as well. So do you think as part of that patient's care that model-informed precision dosing would be in one of your plans at that presentation? So remember that we go back here, we're selecting one drug and a dose that you might want to change. So for those of you sitting back and answering, well, no, at the moment we don't think that might have a role, you can relax, you don't have any work to do. But for those who sort of said yes, I want you to go back and think to yourself, well, which drug out of all that long list of drugs would you choose, and then how would you actually go about it? So that's the end of the first case. Now, most doctors that I certainly know get a bit hot under the collar when we're talking to them about precision dosing. They're going to say, well, I always adjust dose depending on my patient's characteristics. It's ridiculous to suggest that I don't do that. I've got X number of years of clinical experience. I don't really need anyone telling me how to prescribe. But there's a big difference between precision dosing and then our definition of model-informed precision dosing. So what doctors are thinking about when we talk about precision dosing are these factors here. These are these obvious covariants that influence exposure and sometimes influence exposure-response relationships. So of course, we're going to start low and go slow in an elderly lady with mild renal impairment. But what they don't realize is that they're already doing a form of biosimulation. We call that in cerebro biosimulation, otherwise sometimes called modeling and simulation. So together with clinical experience, the prescribing decisions that come out for each patient from that individual is going to be based on those obvious covariants that we know about, plus years of clinical experience. But of course, we now know that there are many, many more factors that are altering exposure and exposure-response relationships, and these are hidden to the prescriber. That's the key word, they're hidden. So it's not a doctor's fault that they've got no idea what to do with a genetic test result when a patient comes in and gives it to them. What does it mean? How do I integrate this information to the patient's care? It's simply not possible to do that using in cerebro, combining it with experience. It needs to be done by more powerful sort of technology. So really for effective precision dosing, the thrust of this slide is we've got to consider all this information and all the information that we know about variants in exposure-response relationships. And this is where I think, in silico biosimulation can be very useful in forming prescribing decisions. So really, I think, the major difference here between precision dosing on that left-hand side, which, we say good doctors are doing every day, and then the model-informed precision dosing, is that, the model-informed precision dosing is going to consider those hidden factors that we know can drive, between subject variability. And, MIPD is really going to be the only form that's going to be able to consider those factors simultaneously in a quantitative approach. So, yeah, yes, that old lady, who's really mild renal impairment, actually, she might be, on the 99th percentile for liver size. So starting low and going slow for her, for a drug that's metabolized, simply isn't going to work, even though you would think based on those sort of known covariants that that's the way we'd have to do it. So, model-informed precision dosing would sort of take consideration of those other factors that almost all of them are hidden to prescribers. Okay, so this slide shows a concept that's not really well appreciated in healthcare, and it's the impact of between subject variability in exposure on efficacy and toxicity, and it comes from Andrew Rowland of Flinders University. We've got 4 hypothetical scenarios, but the top couple of slides here, and also the one down on the bottom left-hand side there's a fairly narrow therapeutic, so there's a very wide therapeutic index for each of those drugs. And the first one here, we've got no real impact of between subject variability on efficacy or toxicity. Here there's an impact on efficacy. Down here there's an impact on toxicity. But as we come over onto the right-hand bottom one, it's a much more narrow therapeutic index. So between subject variability, you can see here, is affecting both toxicity and also efficacy as well. So the premise for any precision dosing strategy is to know the factors that are driving that between subject variability and exposure. And if we know those specific values for an individual patient, then we can use those to predict their actual exposure. And of course, subsequently, if you've got an understanding of these exposure-response relationships, to then have an attempt at predicting response as well. But as I said, this concept of between subject variability and exposure is really, really poorly understood in clinical practice, and most prescribers, don't really apply this at times when it comes to prescribing. Okay, here are our approaches to model-informed precision dosing. The basic nomograms date way back to the early 70s with aminoglycosides. PK/PD models, well, with or without PD. So these are just mathematical models that most people on this call will be familiar with, identifying the statistically significant covariance in a population that are driving, between subject variability. Sometimes they can have semi-mechanistic components to them. So PopPK plus PD, linked to TDM is really the major sort of approach in MIPD used in clinical practice at the moment. So with a base PopPK model, estimating that initial dose based on patient covariance, but once you've actually got a plasma concentration that's been obtained, then you can use that to better estimate the PK parameters for that particular individual, and then predict subsequent dose off the basis of that. So it's a classic sort of Bayesian approach, where new information is used to refine the model. So this is the most commonly used MIPD approach at the moment, and I really think of these as very, very good, dose adaptation tools, once medication is started. So it's a very, very, powerful way of interpreting, TDM data as it starts coming in. So the last one really is, physiological-based PK plus or minus PD. So this is really the basis for virtual twin, which we're focusing on today, where, the models are beginning to incorporate knowledge about biological and physiological functions. So here's a basic PB model, so consisting of compartments that, corresponding to the different organs and tissues of the body with their own permeability characteristics, and of course the compartments are connected with, flow rates that's simulating the circulating blood. And so the PBPK models, providing, once again, estimates of PK parameters, as we would expect, but importantly, you can get, plasma and tissue sort of concentration time profiles out of these models as well. So this is important because we can start estimating concentrations at a site that, may be closer to the molecular target. So if we're interested in central nervous system penetration, then these PBPK models can give us a prediction of exposure in the CNS. So these are not new, of course. If we go back to the origins of PK, this lovely hand-drawn figure by Terrell in 1937, showing how a drug is distributed through the body. So we're seeing exactly the same ideas that we had on that previous slide. Various body compartments connected by blood flow. There's tissue volumes, absorption and permeability rates, and activation rates. So really, at the origins of PK, here we have a scheme reflecting some of that biological and physiological aspects that we now term systems pharmacology approach. So what's really sort of accelerated PBPK is its application in conjunction with in vitro, in vivo extrapolation of a drug's ADME processes. So to do this, you've initially got to separate the systems data and the drug data. So these are the systems data on the left-hand side of the slide, which are really defining the underlying PBPK model. You're bringing out the physiological and molecular values for a particular population. This information is mined from the literature. It's given as a mean, and then there'll be some coefficients of variation around those as well. Of course, then we've got the drug data. The really important one often is the in vitro intrinsic clearance for metabolized drug that can be then scaled up. Of course, the physiological, physicochemical parameters there as well. As part of this, you can define the clinical trial design, but I'm not going to spend any time on that today. But the important thing here is that you marry these two systems area and the data, drug data areas together in a mechanistic IV-IVE linked PBPK model. So this is allowing a bottom-up prediction of those pharmacokinetic parameters. So for example, we've got a drug that's predominantly metabolized by CYP enzymes. we're working out the in vitro intrinsic clearance determined in whatever in vitro system you've got, human liver microbes, hepatocytes, etc. Scaling up using the systems data, such as systems, CYP abundance in the liver volume, going to give you an estimate of hepatic clearance, and this is just one of the parameters that you need to predict PK. So this is all done without doing a clinical study. But of course, importantly, you can simulate the impact of changes to the system on PK. So this allows you to explore PK plus or minus, if you link to a PD model, PK in special populations, it would be very, very difficult to study clinically. So if we start knowing or really drilling down and getting information about how these systems data changes in different states, such as liver disease or obesity, etc., then we're able to predict PK in those special populations or special conditions. And this is what the team at Simcyp have been doing for many, many years. So we know changes in cirrhosis in the liver, of course, changes in obesity, ethnicity. Now there's models for pregnancy, underlying systems data for pregnancy. Pediatrics is very important, and then we're moving into the elderly and even patients with cancer as well. Once you know those changes in systems parameters, and we can apply them to the drug data because we've separated those at the beginning of the process. So importantly, you can sort of, then simulate a lot of these covariants together. So in that elderly lady with mild renal impairment, we can then, simulate what happens if she's a CYP2D6 poor metabolizer, or whether she's taking, multiple CYP inhibitors, and come up with various different clinical scenarios. So this approach, as most of you will realize, has been tremendously successful in drug development. It's gone from academic curiosity to a tool that's very strongly encouraged by regulators to accelerate drug development. And if we look at the new drug applications to the FDA, there's been a massive, rapid increase in the use of BPK studies over the last decade, similar patterns with other regulators as well. So this is mainly in the area of metabolic DDIs, as you can see on this pie chart down the bottom here in the blue, but also increasingly used now for prediction of PK in special populations as well, and taking off in pediatrics as shown in the red there. So the key here is, why not leverage some of that success? Here we are, we've got tremendous success in drug development and applying, these PBPK models, particularly in drug-drug interactions, and now moving into special populations a little bit more. Why not leverage some of that success? So it's been estimated that the team at Simcyp have had over 400 man-years of work gone into the Simcyp PBPK platform. So why not leverage that and try and apply that to a healthcare setting? This is an opportunity, I guess, for a repurposing of that platform. So the virtual twin concept is just very, very simply, adapting some of the systems data and the examples that I showed before in a base PBPK model, just adapting those as best as you possibly can to suit the individual patient that's sitting in front of you that you're going to be prescribing for. So this information is shown in the middle of the slide there. Some of those things will be very, very easy to find, and obviously others will be more difficult to work out. The more those covariant information that then the more realistic your virtual twin becomes. And as you can see, we're going to start to include some of those hidden factors that I talked on before when I compared precision dosing with model-informed precision dosing. So these become fixed. So once you know these values, they become fixed in the model because we know them. And you let Monte Carlo simulation sort of take over and generate some population-level variability around the other system parameters that you don't know anything about. So of course there's going to be lots of stuff that you don't know about, but if you've got a base PBPK model with population variability around those parameters and they're not known for the individual sitting in front of you, then you can let Monte Carlo simulation generate variability around those values, and this should simulate a range of hopefully more realistic PK outcomes. But of course, the outcome is going to be much more better reflective of the individual patient in front of you if you can individualize those parameters as much as you can. Okay, so the overall goal of virtual twins is quite a straightforward concept, but of course, it's a big, big challenge. You want to be providing timely point of care, dosing advice. So the doctor in the clinic or at the bedside already should have, a lot of those patient variables as part of, say, a virtual twin system that we're imagining in the future. Do a mock trial, can be run with the new drug that wants to be prescribed, and of course the key is to spit back some kind of dose recommendation. So for some drugs it's going to be just very basic banding, low, medium, high. More complex medications may need, more fine tuning depending on the dose-response relationships. So you can obviously, come up with different simulations or with silico, different drug doses, schedules, combinations. This technology would allow you to come up with various clinical, dose, sort of scenarios. So the analogy to this is that classic analogy that's been used is that aeronautical engineers are able to generate lots of different designs in silico prior to sort of finalizing the different design that is actually built. So of course that final design would be the drug that's actually given to the patient. So quite a few background slides to go through there, but that sort of finishes off the background section. And we now move into the second section of the talk, which is about virtual twin and the area of psychiatry. And it brings us into our next case study, which is a young gentleman, 19 years old, Mr. BMC, comes in from home. He's actually brought in by ambulance, comes into the emergency department. This is his background, comes from a bit of an unstable, family life. He's presenting with religious delusions. This is a young gentleman who looks like Moses, and he's been flooding the toilets in his local work area, thinking that he's Moses. His medical history is totally different than what we saw with the previous guy in case one. He's on a couple of puffers. So after some workup, the diagnosis, as you can see there, is the first presentation of a psychotic illness. We have a provisional diagnosis of schizophrenia in this young lad, and he started on olanzapine once daily, and he's then admitted to the ward for the full workup. So as we did in the first case, of course, do your management plans or your clinical care plans, would that involve, model-informed precision dosing for this young guy that you're starting on olanzapine at that dose? So while you guys are thinking about that, we'll move through into the proof of concept study for virtual twin that we've just published in the BJCP. Of course, I encourage you to have a look at that and download it, read that after the webinar, but I'm going to go over the key points here. So olanzapine, here are the indications for olanzapine. We chose this as our proof of concept drug because it's a fairly good candidate. It's not metabolized by CYP3A4, so we don't have the CYP3A4 abundance issue to deal with. You can do in vitro and vivo extrapolation of clearance. It's a BCF class 2 compound, so kinetics are not expected to be rate limited by transporters. Therapeutic index has been reasonably well established. Remember, we're talking about psychiatry here. And out of all the atypical antipsychotics, this one's really, really strongly associated with, metabolic issues, weight gain, poor lipid profile. So we thought there's quite a lot to gain from lowering, discontinuation rates on the basis of trying to get the pharmacokinetics right to begin with. So we went back and revisited the in vitro kinetics of olanzapine metabolism, taking into account non-specific microsomal binding and albumin effects. Used a minimal PK model comprising a liver compartment and lump compartment for the other organs. We looked at a lot more enzymes than had previously been investigated, and this work was done up at Flinders University. So here's the paper that published the different enzymes involved. We found that in addition to 1A2, that CYP2C8 had an important role in N-demethylation, and also that UGT1A4, this component was really very significant, so contributing to about 1/3 of the fraction metabolized. So we used that in vitro data, a very thorough evaluation of the in vitro kinetics of olanzapine that we totally redefined, and we used that to build our model in Simcyp. And then we went ahead and validated the model or the compound file in Simcyp against single-dose PK studies, and also a pretty significant TDM database of about 260 patients. In the second part, then we applied the model. So we conducted a small clinical study where we enrolled patients who were commencing on olanzapine. So we created, as I've described before, a best possible virtual twin of those patients with the information that we knew based on the covariants that I've listed here. And the key ones here is we adjusted CYP2C8 abundance based on genotype. We had a few individuals in the study that had genotyping. And we looked at the 1A2 abundance on the basis of the caffeine and paraxanthine ratio. And that was done by looking at saliva samples that were taken for patients. So these are the validation results. We've got the mean fold errors there of the pharmacokinetic parameters, so comparing single-dose PK studies to the simulations in Simcyp. And these are the mean fold errors. Healthy Caucasians, clearance really well predicted, about 18 to 20 litres per hour. Clearance is lower in Chinese compared with Caucasians, down to about 15 litres per hour, and the model performs really well, or reasonably well, in terms of mean fold errors. With the geriatric guys here, the mean fold errors go off a little bit, but the caveat, I guess, for this is that there's much more limited, clinical PK data to make our comparisons. But we thought we were doing a pretty good job on the basis of those single-dose PK studies. Now, these here are the frequency distribution plots of olanzapine steady-state concentration, which are the actual predicted ones in the dotted lines, and then the groupings of the actual concentrations from independent patients, or individual patients, I should say, from the TDN database. The left-hand side, this is 5 milligrams. I haven't got that on the slides. 5 milligrams per day. This is 10 milligrams per day. And on the right-hand slide, this is 15 milligrams per day. So the model is not only capturing single-dose PK, but it's also capturing that steady-state trough concentration, and importantly, that inter-individual variability that we see in our TDN population around 10-fold. On the next slide here, we have the big slide which shows the virtual twin predictions. A and B are the correlations between predicted trough concentration and the observed concentrations. A here is for all 14 patients. We ended up with 14 patients in the study. And then on B, for B, we've got 8 patients who had additional CYP2C8 genotyping. And the correlations are there, you can see on the slide, hopefully 0.833 for all of them, and then comes up to a little bit better with the CYP2C8 genotyping. So C and D are the Bland-Altman plots. There's a slight bias for under prediction if you look closely, but importantly, we're getting right on unity for about 5 of these individuals in the predictions. And importantly, I think we've also picked out this guy down the bottom here where my pointer is just sitting on top because it's quite hard to see. So this is the outlier guy who's really, really low below the therapeutic range. Okay, we then played a hypothetical game about changing doses based on virtual twin simulations or just standard approaches. And the steps I'm not going to go through, you can read that in the paper, but the most important part of this was that after we played this hypothetical game, if you used a model-informed precision dosing approach and adjusted dose on the basis of some of our simulations, then the variability in olanzapine exposure decreases by about 2-fold on the basis of the virtual twin predictions compared to what it would be in clinical practice based on the observed values. So that's described in quite a bit of detail in the actual paper. So go through and have a look at that, and you can see the method that we actually use to try and come up with those predictions. So this is a very powerful slide to present to prescribing doctors or clinical pharmacists to say that some MIPD technology, in this case virtual twin, has the potential to reduce that variability in exposure by quite a lot. And here we've made an estimate of about 2-fold, which we think is really good. Okay, so we move away from psychiatry into oncology, which is the third part of the talk. And as most of you will know, this is the biggest therapeutic area in precision medicine. A lot better molecular diagnoses. There was many novel molecular targets discovered all the time. So lots of new medication, tremendous activity in this area, of course, and the new oral targeted anti-cancer medications, such as the kinase inhibitors, are really ideal candidates for model-informed precision dosing and precision dosing technologies. Okay, so this is our third and final case study for the talk. We've got a gentleman, Mr. BP, so he's a middle-aged gentleman. He's retired now because of his illness. He has a really, really nice family with lots of grandchildren. And he's presenting with a whole lot of nasty stuff going on here, which I'm not going to read out for you. But what I'll tell you is that this is related to his imatinib, which he's been taking for some time for this gastrointestinal stromal tumor. Now, this is unresectable. It's metastatic malignant disease, and he's been on imatinib for about 12 months, initially using sort of toxicity, guided dosing, where it went way up to 400 milligrams BD. And because of some of these adverse events that I've just sort of described here on the slide, having to be down titrated back to 200 milligrams daily due to some of those adverse events. But it's getting to the stage now where these toxicities are really becoming very, very significant and impairing his quality of life. So the diagnosis is, of course, it's intolerance to imatinib after 12 months of treatment for this tumor. But the difficult decision here clinically is that he's got really stable disease. So on the latest in a CT, there's no evidence of disease progression. So we know the drug is working, but of course we've got these toxicities. So the decision is to cease imatinib, and this decision is taken out of the hands. There's no model-informed precision dosing that will do this. This comes from the NCCN guidelines. The guidelines are that for second line for GIST, then you come off imatinib and commence sunitinib. And you can see a very unremarkable medical history with only a couple of other co-medications there. So once again, for our third case, do your plans involve model-informed precision dosing for Mr. BP, this gentleman with GIST? Okay, so these KIs, or the kinase inhibitors, very, very widely studied diverse group of drugs with very interesting basic and also clinical pharmacology. Thank Andrew Rowland from Flinders for these next few slides. These KIs are traditionally dosed using toxicity-guided dosing. So as we saw in the case where you increase the dose, or you get up to sort of top dose and then scale back if you start running into toxicities, because then you at least know that you've got enough drug on board to treat the cancer. But there's accumulating evidence now for PK-guided dosing of these KIs, and the evidence comes from relatively small retrospective studies, and where you're trying to achieve certain concentrations above these sort of steady state concentrations, as you can see on this slide here. So the table down the bottom there is for sunitinib. It's in relation to a metastatic renal cell carcinoma, where you're trying to achieve 24-hour AUC of greater than 800. It's going to give you much better outcomes. I think the value for GIST and sunitinib is around 600. So in our patient, you'd be aiming for a 24-hour AUC of over 600. So I want to talk about some work that's going on at Flinders about Simcyp-guided ADME biomarker discovery, and it's looking at dabrafenib. So dabrafenib is a BRAF inhibitor, and it's used to treat V600 mutated malignant melanoma. And this study that came out last year showed the concentrations above a certain value, so 48 nanograms per mil, were associated with much higher rates of toxicity. So the goal of this project is to use Simcyp, so PBPK platform, to find covariants that are associated with dabrafenib exposures, or could be associated with dabrafenib exposures greater than this value of 48 as found in this paper. So at Flinders, there's a fantastic suite of full PBPK compound files for a lot of kinase inhibitors. They developed one for dabrafenib, and this slide here shows the verification, which was done using the Genentech cancer population file. So the systems parameters in terms of the verification were based on that previously defined population file from Genentech. And there are a couple of new population files for cancer patients that are out at the moment as well. And then they used the FDA guidance to do best practice PBPK model building. So they used a univariate logistic regression analysis to screen associations of the patient characteristics in Simcyp. So we're talking about all the physiological parameters that drive the software plus the molecular characteristics, to see if there's an association between any of those characteristics and a steady state concentration above that value of 48. And then using multivariate logistic regression model and back extrapolation, deleting those baseline characteristics that were significant in the univariate model, they assess predictive performance using the ROC AUC curve as shown on the right here. So the outcome of all this is that the real simple sort of take-home message for it is that, you get a really good, if you consider baseline weight, CYP2C8 abundance, CYP3A abundance, and also PGP abundance, plus, as I said at the beginning, the patient weight as well. All those covariants describe between subject variability and dabrafenib exposure really well. And there's actually a very, very strong, predictive performance for steady-state concentrations, predicting steady-state concentrations above that value with an AUC of 0.94 and an accuracy of 88. So I'm told these are really, really good values. So if you had information about those covariants, you just need to use your imagination. If we have information now about those covariants for an individual patient, of course, the next step is that you'd be able to predict, a priori, the risk of toxicity at standard doses, if you understand what those abundances are for an individual patient. So the next step as part of this project will be then to apply this clinically to predict those that are at risk of toxicities. Okay, so that's the final case in the section about oncology. Just want to briefly go through some of the potential advantages or the advantages that we see of virtual twin, the virtual twin approach, certainly that's been developed by Certara. Some of these are quite straightforward. The PBPK platform that we have, the Simcyp is very, very user-friendly interface, so you don't need to have extensive training in pharmacometrics. So it's going to be able to be used by clinicians like me, which is kept nice and simple for us. You don't need a lot of pharmacometrics. The other thing is we see that it can be a rapid application to novel clinical scenarios. So there's no need to generate an individual model each time that's only going to be valid for a certain population that you're looking at. So for example, if you had a validated new drug that had been validated in healthy volunteers, and then you wanted to apply that to an ethnic population, with that ethnic population had previously been good at predicting its PK for a whole heap of other drugs, and of course you'd be able to simply combine those two and have fairly decent confidence that your PK estimates would be okay. So rapid application to novel clinical scenarios or clinical situations we see as a potential real benefit or advantage. Flexibility to change. So once you're beginning to bring in information about the in vitro data for disposition, if you're adding new interacting drugs into the system, these systems have great flexibility. Also, you have the ability to be able to, simulate intrinsic or extrinsic factors, outside the range that have been found in the clinical data that perhaps have been used to build another type of model, such as a PK/PD model. So this would allow you to have, the flexibility or better assessment of, what-if scenarios clinically that actually may occur in clinical practice. The other thing that we're excited about is, the possibility, of having some clinical scenarios that could save time in hospital or outpatient clinic. So the potential cost savings of virtual twin technology, if we choose high-impact model-informed precision dosing and develop the evidence base for that. So we're quite excited about that area. So this comes from the Darwich and Armin Rostami state-of-the-art paper from CPT last year, which is a brilliant review of model-informed precision dosing. And what it does is it describes the patient characteristics. I'll let you guys read some of those things that are dot points there. Obviously the drug characteristics as well, and the disease characteristics that we think are the prerequisites for high-impact model-informed precision dosing. This point down here is it should actually be identified relevant biomarkers to drive dose selection. So this is all one dot point down there on the left-hand side. So what we're doing at the moment is we use this as a really good framework for identifying potential areas of clinical implementation. We're not naive about model-informed precision dosing. We realize that it's going to be applicable. We need to be very, very selective about the areas of clinical medicine where we start to focus on, where virtual twin technology could have the most impact. And we look at this as a framework where that might be the case. So here are some examples here. There are many, many clinical scenarios that I've been able to come up with where getting that initial dose correct could save quite a bit of time and quite a bit of money, and I've listed 4 scenarios here. The first one, for example, in treatment-resistant schizophrenia. So those of you who know anything about clozapine will realize this is a narrow therapeutic index drug that's quite difficult to dose in clinical practice, and dose initiation is typically done according to a very standard initiation protocol over about 2 to 3 weeks, because what you're really worried about here is overshooting the mark and getting toxicities, such as seizures or neutropenia or very, very severe sedation. But the trouble with this is there's large individual variability, as with a lot of psychiatric drugs, in therapeutic concentration. So quite a few patients will be right at the bottom end of that range, and they can actually be subtherapeutic for very long periods of time and end up spending a lot of time in hospital while we're waiting for responses and waiting for some TDM data as well. So prediction of that initial dose before starting could allow what we would say would be a more personalized, perhaps a more rapid titration schedule for such patients who are predicted to be way down on the lower end of the scale. And of course that could actually decrease the number of non-responders that are simply due to pharmacokinetic issues. So we have a trial beginning at the moment in this area where we will see whether model-informed precision dosing that picks out a final plasma concentration of clozapine predicted at steady state, and we let the psychiatrists have that information, and then they can compare that a personalized sort of titration strategy for those individuals versus the normal initiation protocol. So we think that these type of projects, and I've listed some other examples there are a lot of other examples in clinical practice that I could have listed, but there's some other ones there. Paroxysmal AF, there's treatment of acute pain. A lot of people sit in the hospital with pain that's very poorly managed initially, and we've talked a little bit about oncology and hematological malignancies with the kinase inhibitors. So we think that getting that dose right first up could translate into decreased length of stay in hospital. As I said, in psychiatry, this could be weeks of costly time. So the ambition here would be, if model-informed precision dosing could be applied in those high-impact areas, then we would have the ambitious target of reducing some of that hospital time and that outpatient clinic time. And we would like to set a target that's very ambitious, but wouldn't it be an amazing sort of impact or a huge sort of global impact on healthcare costs if you could reduce that time or that length of stay by about half in very, very select therapeutic areas or select cases. So very much, marrying a lot of information about the drug, the disease, and also the patient characteristics as well, choosing those high-impact model-informed precision dosing areas, and then, applying it and trying to generate evidence in those areas. Okay, so I'm coming towards the end of the slides. Just in conclusion, we had 3 clinical cases that I went through in a bit of a hurry, but the first one was about complex comorbidities. So for me, you guys will think about what your plans were, whether they involved any potential MIPD technologies. So for me, in that case, I've got some higher immediate priorities, to be honest. This guy's got some critical limb ischemia, which is a medical emergency. So as part of the initial management plan for that individual, at the moment, I have to stress, at the moment, the initial management plan, MIPD, is not going to be part of the plans at the moment. But this is an exciting opportunity, I guess, in the future, maybe solving some of these very, very complex polypharmacy sort of dosing questions could be an area as this technology, expands and becomes more reliable and the evidence base is generated. So for that particular case, I would have perhaps picked out rivaroxaban as the drug, or maybe even metformin as a couple of drugs that may be candidates for model-informed precision dosing in the future. I think case 2 with the new psychotic illness, even though psychiatric sort of endpoints are very dirty, look, this is a great case where estimating exposure initially could be really beneficial. So, as we saw on the virtual twin proof of concept paper, if you identified the outlier patient, and this is the guy who's sitting around in hospital chewing up lots of healthcare resources, so they've got a very low concentration, and if we could identify that patient early, then this would be, on the basis of PK issues, of course, at the moment, not the pharmacodynamic endpoints, then this may have some, really, really positive potential. And I think case 3 really is, I think it's a beautiful case where, MIPD, could have massive, clinical impact really and improve the quality of life for an individual. So you need to get them onto a different drug, get that dose right, quickly, maintain efficacy, but also, try and address, with the view of trying to address all those really, really nasty toxicities that individual was suffering. And you've got to remember for that patient, this is the last phase of their life. And, I think there's a really, massive sort of positive potential of MIPD for such a clinical case. Okay, the next thing then was on the slide, we have just the proof of concept study. I went through that with you, virtual twin that we recently published. And also we looked at the cool stuff that's been doing at Flinders with Simcyp ADME biomarker discovery in the area of oncology in the dabrafenib paper, which is soon going to be coming out in CPT. And then I think in conclusion, repurposing PK platforms that are already out there is an option for MIPD. And I think it's going to require quite a lot more work to examine the clinical potential, but it's a really, really exciting area and something that I'm really looking forward to. Okay, so just some quick acknowledgements. I'd really like to highlight Andrew Rowland from Flinders University, who's here in Adelaide. He's been involved in most of this work, but also my colleagues at Certara as well, Craig, Armin, and Masood. So I'd be really delighted to, I know I went through those slides pretty quickly. There's a lot of information in there, but I'd be really delighted to answer any questions now. And of course, we do have limited time. So those of you who have questions and have typed questions in that I don't get time to respond to them, please, send me an email with your questions at my Certara address, and it'd be fantastic to, connect via email if I can't get to those questions today. So I'll hand back to you, Suzanne. Thank you.
Thank you, Tom. As Tom said, we encourage our audience members to submit his questions to Tom in the Q&A box. Looks like the first question, someone asks, for building the base virtual twin model, it's a two-part question. Do you recommend including demographic data such as age, gender, glomerular filtration rate, et cetera, into account when you compare it to the observed data? And then the second part is, can you talk about the model verification processes such as you would use for PBPK in drug-drug interaction studies?
Yeah, so the first part of the question, Suzanne, so we would try and incorporate, all the information that we have about the individual patient. So I think it's all relevant. So, the base demographic data, of course, age and weight and all these things are very, very important. So the more of that information that you have, the better. And if you've got a sense of, renal function as part of that as well, that would be incorporated into the virtual twin model. So really the aim here is to, if you know that information and even though if you're suspecting this is not going to be an important covariant that's going to just, describe some of that between subject variability, I would still really include that in the model to try and, mimic the actual patient as much as you possibly can. So the answer to the first part of that question is yes, you'd be including as much information that information as possible in the virtual twin model. The second part, of course, is we'd just be following now the standard FDA guidance about verification and development of those models. So I think a really nice example of that is, there's obviously lots of papers out now that are beginning to do that, which you can see examples. But the dabrafenib work that's been done at Flinders is a really, really excellent example of that, where just following step by step the FDA guidance on development of the model, and that dabrafenib model, as I hopefully showed on that slide, seemed to recapture very, very well using the Genentech cancer population. So yeah, it's definitely following those standard FDA verification pathway.
Someone would like to know, can you speak to some of the challenges or difficulties and what sort of infrastructure or personnel would be needed to help deploy model-informed precision dosing technology in healthcare settings?
So there are a number of challenges, of course, like everything. Incredible opportunities in this area, brought about the challenges, in relation to the challenges. So, for example, the science of in vitro, in vivo extrapolation, a key factor that I see as being really important in moving this technology forward will be to have some non-invasive ways of picking out CYP abundances. Of course, CYP3A is absolutely critical, so a non-invasive way of getting that information would be very, very important. There's a lot of, I think, thinking that needs to be done about, the top-down, marrying top-down with these bottom-up approaches and having some structure around that. How much, do you inform the models, and how many of these system parameters need to be, estimated, and some more structure around that as well. So the challenges there's lots more challenges in terms of very simple stuff. If we park the science aside, then there's very big challenges in just the culture of prescribers and the culture of say hospital settings where the priority for a lot of people doing the bulk of prescribing isn't necessarily they're not clinical pharmacologists or people who are really, really focused in on drug dosing. I said in one of the slides that between subject variability and exposure is fairly poorly understood, I think, in general in healthcare. So there are a lot of educational challenges around educating individuals, prescribers, certainly at the junior level, about some basic clinical pharmacology concepts, because if they don't realize that there's a problem with such variability leading to poor outcomes, either low efficacy or adverse drug events, then if they don't realize there's a problem, then there's going to be no impetus for change, and there'll be no recognition of whether these model-informed dosing technologies could be utilized. So there's the scientific challenges, and then there's education challenges, which are very important as well, which we're working on. Then I think the second part of that question was in regard to who do we see leading this type of, the push, or who would actually be the end user and applying these technologies in healthcare. So I think it's a collaborative effort between prescribers, but I would really feel as though clinical pharmacy would be absolutely essential in driving, this sort of technology along, where, decisions to prescribe and drug choice is left to the prescriber, where it's fine-tuning or initiation doses, and then fine-tuning of doses could really fall under clinical pharmacy's area. So I think it's going to be a collaborative effort, but I would really love to see very, very high-level engagement from clinical pharmacy as a profession. And at the moment, certainly in Australia, that the ability to be able to make dosing recommendations is not very strong from the clinical pharmacist. So I'd love to see this technology pushed along by them.
Will the clinical study planned for olanzapine use the virtual twin technology to decide the dosing for individual patients?
So the twin and the validation, it hasn't been applied to actually predict dose for patients. So it's really just the proof of concept study was there to see, can we predict steady state plasma concentrations in those guys? And now that we've done so and we've developed that model, of course, naturally the next phase would be then to apply that model and give that information to clinicians in order to guide dosing. The trouble with olanzapine in our institution at Flinders is that it's used as part of the acute agitation protocol. So it's quite difficult which can actually vary with PRN dosing. So it's quite difficult to find individuals who are being started. This is why we only had 14 individuals in that small clinical study. So it's quite hard to find individuals who are just starting off olanzapine without having had it as part of an acute agitation protocol. So we feel as though a better place to start a sort of a clinical impact study would be with clozapine, and that project is advancing quite nicely. And I think, as I said during the talk, so developing the baseline model for clozapine is pretty well advanced, and the study will compare, or giving psychiatrists that extra information. It's not to say this is the dose we want you to prescribe, it's to say here's our normal initiation protocol for clozapine. Half will follow that, and then the half will get the actual steady state plasma concentration that would be predicted for the individual, and then we'll leave it up to the psychiatrist to determine how they use that information to a degree. So we haven't applied the olanzapine model, but we're certainly looking at applying the clozapine model, and hopefully that reveals some really exciting data in the future.
Someone is asking about, do you feel that there are any conflicts with the use of patient personal data for making these virtual twin simulations?
Well, I think most of a lot of the information is part of electronic health records as it is at the moment. So you're going to be getting all those basic stuff that we talked about, the age and the weight. So there's no issues there. Liver function tests and renal function, some of these things are obviously available. There will be issues, I guess, around when individuals have had their drug metabolizing enzyme or transported genotypes, and how that information becomes available and how it's utilized. There'll be things, the drug-drug interaction side of it will be, obviously, transparent because it's going to be part of their, e-medication list. It's really going to be around the issue of, the genetic testing for some of those DM ETs that are genotypes and how that information is utilized. So there's a whole lot of issues there that I probably won't go into now, but I think it's going to be very interesting to have a framework for how that information could be applied within the virtual twin. And I guess it's going to be up to maybe certain individuals are going to release that information to populate their virtual twin with that information if they so decide. But certainly in Australia, where genotyping is very, very rare at the moment for those enzymes and transporters, there'd be more individuals who have gone off and got that information. I'm sure they'd be probably very keen to let that information be used as part of a model-informed precision dosing strategy. But there's a lot of issues around there that's quite interesting. So that's an excellent question.
Okay. When physicians use model-informed precision dosing tools in their clinical practice, how can they be sure that the virtual twin generated by the technology actually matches our clinical patient?
So this is where we're starting to build the evidence. It's going to be, a lot of effort over the next few years. It's going to be going into, various areas of clinical medicine to try and, build the evidence. Initially, it's going to be from an observational point rather than an interventional point. You need to build the evidence for, as we saw with olanzapine, whether you are able to get really decent predictions of final steady state concentrations, focusing on PK, of course, initially. And we've targeted at Certara together with academic partners, have targeted 5 areas that we're interested in, and we've got partners with. Manchester University, we're looking at renal impairment. At Geneva, with the Jean-Luc René, we're looking at a lot of the DOACs and cardiovascular. Steve Leder in Kansas City Children's Mercy Hospital has got an atomoxephene study. We're partnered with Florida in the area of opioid addiction, and partnering with Flinders here in Adelaide in terms of oncology and also some psychiatry as well. So those 5 sort of key academic centres that are embracing this technology and developing virtual twin technology is going to be generating, I guess, that evidence to try and, we have to sell it in terms of evidence-based medicine concepts to prescribers. So it's going to be, building that evidence through those programs.
Looks to be the last question from our audience. What would you say is the applicability of virtual twin in the clinic in the context of a new patient regarding the time that's required for the simulations, the analysis, that sort of thing? Yeah,
so initially, I think it's going to take a little bit of time to generate these individuals. You would need to have, a PBPK platform and a virtual twin platform that you can input data prior to patients actually rolling up at clinic. So in actual fact, you're going to have virtual twin of that individual before they come to clinic already developed. So, initially it'll be time consuming, but as time goes by and the evidence base builds and people become more familiar with this type of tools, and of course that time would become smaller. At the moment, you see it being in very, very focused areas of clinical medicine where there are strong where you build evidence for strong longer term outcomes. So, for example, oncology, where that individual is switching on to sunitinib, really the time isn't a great factor in that case because you're just going to spend the time putting together the virtual twin for that patient because it's absolutely critical. Whereas if you're in a more acute setting, of course, the time of putting together a virtual twin and the potential benefits of that approach would be much more limited. So it's a matter, as I said, of identifying those high impact areas where the extra time that it's taking to put together a virtual twin and come up with some predictions is actually very, very valuable in terms of the outcome for that patient. So acute settings, of course not, but chronic, longer term conditions such as in oncology, identifying those high impact areas of MIPD will be very, very critical.